All right, folks, welcome to our Zoom meeting. We're going to be kicking off in just a couple of minutes, but for now we've got the agenda pulled up and you can take a good look at it. My name is Leah Cuddyback and I'll be kind of helping to facilitate things here. So if you've got any questions that come up or concerns, you can feel free to drop them in the chat. We're going to go ahead and let this meeting run with everybody having the ability to kind of see who's present in the room. We ask that folks keep their microphones muted until the end when we're doing a question and answer section, and we'll at that time have some more opportunity for conversation. But for now, welcome and stay tuned. We'll be kicking off shortly. All right, folks, and we've got about 3 minutes until we're right at 12 and we start to kick off. And at that time, we're going to have some of our panelists kind of turn on their cameras and start talking. But for now, just to let you know, you're in the right place. We're going to be getting started right at 12 with a little introduction from Jason Pinchback with the General Land Office.
Thanks, Leah.
And I will. Yeah, go ahead, Jason. What's up?
Oh, thanks, Leah. I was just going to ask folks if while we're waiting here in the next few minutes, if you would like, please use the chat function to introduce yourself, your affiliation, or your area of interest along the coast. Leah, sorry to interrupt. I didn't know if I cut you off there.
No, that's totally what I was going to say too. I was also going to let folks know, right now I've seen a lot of screens. We'll have folks having the opportunity to turn off or on their cameras, and we'll have some opportunity for more interaction at the end. But for now, thanks everybody for tuning in. And Jason, I have a feeling we're going to have folks filtering in a couple minutes after things start kicking off. So I'd say let's not worry too much and let's go ahead and do our intro. We're right about 12. Seeing about 55 folks here already, but I know we've got about 130 registered at this time. So we'll stay tuned. And thanks to everybody that's tuning in. You're in the right place.
Absolutely. Thank you, Leah. My name is Jason Pinchback. I'm the Water Resources Manager at the Texas General Land Office. Thank you so much for joining us today. We're super excited to launch our inaugural Lunch and Learn series. And the Lunch and Learn series is really meant to share knowledge and experiences about coastal water quality, and to talk about issues and to generate discussion and ideally build interest in maintaining and restoring our estuaries, beaches, bays, bayous, creeks, streams, springs, and lakes in our coastal zone. The Lunch and Learn series is part of the Clean Coast Texas program, which is an initiative involving several state agencies. And what we're trying to do is to work with communities to ensure vibrant and sustainable fisheries, shellfish, and ecotourism industries. Through planning, constructed improvements, environmental analysis, technical resources, and partnership activities that ideally work to enhance water quality management. The Clean Coast Texas program is guided by the Texas General Land Office, the Coastal Management Program, in partnership with numerous stakeholders, including state agencies and other local agencies. Resources provided by Clean Coast Texas can help coastal communities reduce environmental impacts of stormwater runoff from existing and urbanizing areas, and also enhance wastewater treatment. And our overall goal is really to support a thriving Gulf Coast economy here in Texas, as well as an environment, of course. Whether you're a city manager, a developer, a homeowner, business, a nonprofit organization, or a public servant, we think that there's a role for you in working with us in protecting coastal water quality in Texas. Before we move any further, I just wanted to mention a couple of housekeeping issues. A lot of folks might have questions or comments, and we're going to ask you if you could please use the chat function. And we have about a 30-minute presentation here in a few minutes. And we will, at the end of that, we will then address comments and questions to the end of the hour. And so if you could please hold on to those. Everybody should be relatively muted or totally muted throughout the presentation today. And we'll have, again, we'll be able to address your comments and questions at the end. If you haven't already done so, please use the chat function to introduce yourself, your affiliation, and or your area of interest. Today, I'm super excited that we have Ms. Nicole Powers, who is a PhD candidate at Texas A&M University-Corpus Christi, to share some information about data analytics and water quality for Texas coastal beaches in Texas here. Just to give a brief background of kind of how we got here and what we're doing in regards to looking at water quality from a General Land Office perspective, is a couple of years back, the Texas Beach Watch Network, which is managed by Ms. Lucy Flores and our NOAA Coastal Fellow, Brian DeSanti at the Land Office, observed several months of very high and anomalous bacteria readings in two particular counties along the coast. These very high readings were concerning to us and stakeholders. We had a lot of communication from local affiliates. And the General Land Office initiated an investigation to try to understand and characterize what was causing the high bacteria readings. And so we spent several months looking at the issue, and actually quite a bit of time over a period of a year, in which we began to develop some theories on what was going on and why it was going on. But we also understood that the mechanisms that impact water quality in the coast are very complex and very vast. So we realized we needed some more capacity, expertise, and the big data guns, if you will, and started working through partnership with Dr. Jeff Turner, Dr. Mike Wetz, and eventually bringing us to forming a small team with Nicole Powers being a big essential part of that. And so without any further ado, what I'd like to do is introduce Ms. Nicole Powers, PhD candidate from Texas A&M University. Thank you so much. Nicole?
Great. Well, thank you for that introduction. Let's see if I can share my screen. Here we go. Okay, I think that's working now. All right, well, thank you again for that introduction. And today I'm here to talk to you about some of the water quality in Texas beaches along the coast. I'd like to start off with some background information. You may be familiar with the issue of fecal contamination that can impact coastal waters, especially here along the Texas coast. These are pictures of a few popular news articles that talk about the fecal pollution that's present in the water. On the left here, we have your annual poop alert for visiting the Texas coast. And there are a couple more articles listed here. And you may have seen some of these yourself if you're visiting the Texas coast, planning a vacation. And in the bottom right-hand corner here, we have a sign for the Texas Beach Watch program, indicating that the beach where this sign is located is being monitored for bacterial pollution. And fecal pollution in the water can be particularly detrimental. It can consist of pathogens that can cause infection in humans who interact with the water. Say if somebody is going swimming and they accidentally ingest some of the contaminated water, or if they have an open wound, even a small open wound that comes in contact with pollution in the water, that can also lead to infection. One of the ways in which we can monitor water quality is through the measurement of fecal indicator bacteria. So these serve as a proxy for fecal pathogens that can cause disease, and those pathogens that are present in the fecal material and fecal waste that is accidentally let out into the water. You're probably familiar with E. coli, which is an indicator bacteria that's used typically in freshwater settings and for drinking water. However, Enterococci are another example of fecal indicator bacteria, and they are typically used in marine settings, so where there's salt water. Numerous studies have shown in the past that as the concentration of Enterococci in the environment increases, so do the number of illnesses reported in people who have been exposed to that water or have been interacting with that water. And this is particularly true in locations that are impacted by a known point source of pollution. So I'd like to give you a little bit of information about Enterococci here. On the left, we have a picture of Enterococci. Each of those blue dots is a colony forming unit. And these, like I said before, these are the preferred marine water quality indicator. However, they're not host specific. So that means they can be found in the presence of the gut and fecal material of humans, as well as other warm-blooded animals, including birds and dogs and mammals. And they can also be found in the environment to an extent. They have been approved by the EPA, and they have been Texas's standard for water quality since 2004 in marine recreational water. And the Texas Beach Watch program routinely monitors levels of Enterococci at certain sites along the Texas coast. This is a map showing those sites that the Texas Beach Watch program monitors. So this is a screenshot I took a couple weeks ago from texasbeachwatch.com. And basically each of these dots on this map represent a sampling station. And the green dots are locations where there were fewer than 35 colony forming units of Enterococci per 100 milliliters of water. And the yellow dots represent sites where there were between 35 and 104 colony forming units of Enterococci. And there are no red dots on this map, which is a good thing. But if there were red dots, those would represent sites where there were greater than 104 colony forming units of Enterococci. And I checked again this morning, and the vast majority of the sites are green, so that's good news. And so this 104 colony forming units, that's kind of it's the beach action value set by the EPA. So when you have Enterococci present in the environment above that concentration, there's an associated elevated health risk with humans that interact with the water. So this is kind of a magic number that we'll be coming back to a few times in the presentation. And for marine recreational water, you want there to be fewer than 104 colony forming units in the water. So that brings us to the purpose of this project. Our goal was to characterize fecal bacteria pollution across coastal Texas. And to do so, we had three main objectives. The first was to analyze historical Enterococci data, and we wanted to rank the beaches and coastal counties based on their water quality. And finally, we wanted to test for correlations between Enterococci and relevant environmental variables that might be contributing to elevated fecal indicator bacteria levels. So all of the water samples were collected and analyzed by the Texas Beach Watch program. Water samples were obtained from a total of 10 coastal counties. which included 66 beaches, 169 sampling stations, and the data analyzed in this project was collected for over 10 years, from 2009 to 2020. And we had over 75,000 individual water samples that we analyzed and were working with. And here's a map, again, just reminding you of where all of those sites are located on the Texas coast. So in order to measure the water quality, they quantified Enterococci, and they did so using the Enteralert test method. And I'll briefly tell you what this entails. So this test involves a nutrient indicator that you mix in with your water sample, and then you pour it into this tray that's pictured here in this photograph, and you seal it up and you incubate it overnight. And the next day you can go in and see what has grown. So if Enterococci is present, it will fluoresce under UV light because of the nutrient indicator that's present. And based on the number of fluorescent wells, you can determine your most probable number, or MPN, of Enterococci per 100 milliliters of water. So moving forward with this presentation, when I am talking about Enterococci, these are the units I'm referring to, MPN, or the most probable number, based on the Enteralert test. So that brings us to the results of this project. Here we have a table showing the summary statistics from all of the samples that were collected, and I'm going to walk you through this table. Here we have the county and the maximum value of Enterococci that was detected throughout this entire study in each county, as well as the median value and the average value of all of the samples collected within each county. And then the number of beaches that are present in each county that we tested. And then finally, over here, we have the percent exceedances. And these are the percentage of samples that exceeded the beach action value of 104 MPN. And based on this number, the percent exceedances, we were able to classify each county within one of these categories of having low, medium, high, or very high levels of Enterococci. So in green, we have the low levels, and then yellow, medium, orange is high, and red is very high. And this pattern, this color pattern, will follow us throughout this presentation. So here's a map showing you exactly where these counties are located. Again, the color coordination matches the previous slide. So we have Cameron and Jefferson, which had low levels of Enterococci that exceeded the beach action value less than 5% of the time, as well as the yellow counties, Galveston, Aransas, and Kleberg, which had medium levels of Enterococci, and the orange counties, Brazoria, San Patricio, and Nueces, had high levels. And then finally, Matagorda and Harris had very high levels of Enterococci that exceeded the beach action value in over 20% of the samples. So finally, we wanted to look at potential drivers of the Enterococci levels that we were detecting throughout the study. And one of the things we wanted to look at was location. So as you saw in the previous couple of slides, location did have a big role to play in this. Certain counties had much higher levels of Enterococci than other counties. And in addition to the counties, we also wanted to do a comparison of Bayside versus Gulfside sites. So Bayside sampling sites were located directly on a bay, and they represent low flushing environments with high residence times, where the pollutants that enter that water might stick around for longer. And we compared those to the samples obtained from Gulfside sites that were located directly on the Gulf of Mexico. And as you can see, there is a significant difference between the average Enterococci concentrations on the bay. In fact, the average concentration was over 3 times higher than the beach action value. However, the Gulfside sites had significantly lower levels of Enterococci that were well below the beach action value of 104 MPN. In addition to location, we wanted to see if there were any temporal trends in the elevated levels of fecal indicator bacteria. So we wanted to see if they were increasing or decreasing or just staying the same over time. And here we have a graph showing the percent exceedances of the beach action value, so that 104 MPN, throughout the entire study in 2009 to 2020. And you can see a definite positive linear relationship between these two variables, which indicates that there are more percent exceedances in recent years than there were, say, 10 years ago, which, let's see here in this slide, this shows approximately the same thing, but instead of looking at the beach action value, we're looking at the upper limit of detection for the Enter-Alert test, which is greater than 24,196 mpm, quite a bit higher than the beach action value. But the trend here is the same. We see in recent years, there are several more occurrences of enterococci concentrations that exceed this upper limit of detection. Two important variables that were correlated with time are population size and sea level. In the majority of counties, the populations have been increasing and sea levels have also been rising. So we wanted to investigate these two factors. So this is a table showing the correlation between enterococci and time, population, and sea level in each of the 10 coastal counties. And there's a star here by Kleberg County. I wanted to let you know that the samples from this county were only obtained through 2011, which could explain why there are some non-significant relationships here with these tests. But the rest of the counties had data through the year 2020. And there's a lot of numbers on this table, but what I want to point out to you is that the largest numbers are towards the bottom of the table in the counties that have the most impaired water quality already. And if we look at Harris County, for example, this county experienced the strongest correlations between enterococci and sea level and population growth and time, which implies that the water is going to continue getting worse as time goes on and as sea levels continue rising. And some of these correlation values are relatively low, which is to be expected with environmental data such as this, because there are so many different factors that can influence and impact the levels of fecal pollution in the water. Here's another figure showing basically the same thing as the previous table, but in a graph form. So on the y-axis, we have our percent exceedances of the beach action value. And we have correlation between enterococci and time, population, and sea level. And again, this is showing there's a clear positive relationship between these factors. So I wanted to give you an example of one way that sea level can impact water quality in coastal regions. This figure was taken from Elmer 2018. They did an analysis of thousands of septic systems within the state of Florida, and they determined that sea level was impacting the septic systems and contributing to impaired water quality. So in order to function properly, septic systems need to be surrounded by unsaturated soil. The septic tank and the drain field need that unsaturated soil around them. However, as sea level is rising, groundwater is also rising. And this groundwater can encroach on those systems, and they can become compromised, or they can even fail when they become submerged with this water. In addition to sea level and population size, we also wanted to analyze rainfall and the occurrence of sanitary sewer overflows and their impact on the water quality. And rainfall has been associated with high levels of fecal pollution in numerous studies in numerous locations in the past. And I'd like to show you this video here, All the Cups, that was made by Jace Tunnel. This was recorded after a 1-inch rainfall event that occurred in Corpus Christi. So this is Corpus Christi Bay, specifically Cole Park. You can see that a massive amount of stormwater runoff was produced from this rainfall event. I will play this again. And all of that stormwater runoff was let out directly into the bay. You can see all of these cups and all of the trash that was part of the stormwater runoff. But what you can't see are all of the microscopic pollutants that are also accompanying all of this litter and trash and those styrofoam cups. I'll play this one more time. And even smaller rainfall events that aren't nearly as big as this one or don't produce as much stormwater runoff can still contribute to elevated fecal pollution in the bay. And I also want to mention sanitary sewer overflows. Storm systems and sanitary sewers are designed to be completely separate. So a sanitary sewer system is designed to take waste and send it directly to a wastewater treatment plant where it's treated before being released into the environment as treated effluent. However, separate storm systems, the storm sewers, they collect all of the stormwater runoff and let it out directly into the surrounding environment or the bay, instead of taking it to a wastewater treatment plant for treatment first. However, sanitary sewer overflows can happen in the event of large rainfall events, or if there's any sort of a blockage in the sewer system. And even small cracks or large cracks and breaks can contribute to fecal pollution as well, especially when there are leaks in the sanitary sewer systems and then rainfall occurs and flushes out the system and takes that untreated sewage and waste and deposits it directly into the bay instead of taking it to the wastewater treatment plant first. So we had a partial list of known sanitary sewer overflows that occurred in Brazoria County from 2018 to 2020. And this graph is showing all of those dotted red lines correspond to a date of a known sanitary sewer overflow. But like I said, this is a partial list. So there were other occurrences that were not captured within this study. And all of the black dots on this figure represent a water sample and the concentration of enterococci within that water sample. So you'll notice that the red lines sometimes coincide with elevated levels of enterococci. However, that's not always the case. So clearly there are other factors that might be contributing to elevated enterococci levels. If you notice here, there's a large peak in enterococci concentrations in 2019. And this is what Jason was mentioning earlier at the beginning of this meeting. So we wanted to see if sanitary sewer overflows could have been one of the things that was responsible for the elevated concentrations detected throughout 2019. And it's possible that they are, but it's clear here that they're not the only thing that would have been contributing to elevated fecal pollution during that time frame. So I want to give you a closer look at those concentrations we detected in 2019, particularly in Brazoria, Matagorda, and Harris counties. And here are the beaches that were tested within those counties. What I want to show here is a comparison of the data collected in 2019 to the data collected throughout the remainder of the study. So 2009 to 2020, excluding the year 2019. All of the rows highlighted in red represent the data from only 2019, and the unhighlighted row above it represents data from the rest of the study. So you can see all you have to do is compare the unhighlighted row to the red highlighted rows, and you can see a massive difference between the percent of samples that exceeded the beach action value. If we look at Harris County and Sylvan Beach down here at the very bottom, almost 60% of the samples exceeded the beach action value in 2019, whereas throughout the rest of the study, it was only 18%, which 18% is still very high, but there's a very big difference between 18 and 60% of the water samples that exceeded the beach action value. So like I said, this is the same slide as before. We wanted to see if sanitary sewer overflows could be contributing to the elevated concentrations of enterococci, and it's possible. However, there are likely another common set of factors that might be contributing as well. The fact that all three of these counties experienced elevated concentrations in the same time frame means that there was probably a common set of conditions that was influencing the bacteria levels. And rainfall and sanitary sewer overflows definitely could have been some of those factors, but they were definitely not the only thing contributing to the elevated levels. So I'd like to go over some conclusions and take-home messages from this project. We saw that location had a significant impact on the concentration of enterococci in the environment. Specifically, certain counties had higher levels of enterococci and had more occurrences of exceedances of the beach action value. And we also saw that Bayside sites had much higher levels of enterococci that also exceeded the beach action value compared to Gulfside sites. We saw that enterococci increased with time, and in particular, more than 60% of the beach action value exceedances occurred within the last 5 years, and almost 90% of the exceedances of the Enterolert test's upper limit of detection also occurred within the past 5 years. We saw that enterococci were also increasing with population size and sea level, And this is a little bit worrisome, particularly in the coastal counties that are experiencing population growth right now, and the ones that are vulnerable to future sea level rise as well. And finally, we saw that 1/3 of the beaches, which included half of the counties, exceeded the beach action value on a regular basis, or more than 10% of the time. We have some future plans for things that we would like to continue investigating. We want to learn more about the elevated levels of Enterococci and see if there's another occurrence like there was in 2019 in the future, and what might be contributing to those elevated levels. We'd like to look into the possible sources of the pollution. For instance, I mentioned rainfall and sanitary sewer overflows, but also septic systems. It would be very informative if we could investigate the age or the maintenance or the type of septic system that might be in a coastal region to see if those are contributing to elevated levels as well. And finally, we also want to do some bacterial source tracking. I mentioned earlier at the very beginning of this presentation that Enterococci are not host specific. So that means they can be found in the gut and fecal material of humans as well as many other animals. So when we find or detect elevated levels of Enterococci in the environment, that's an indication that there might be fecal pollution present. However, it doesn't tell us where it came from. So with bacterial source tracking, we are able to look for genetic markers that are specific to the gut microbiome of humans or specific to the gut microbiome of other animals, such as dogs or gulls. And once we identify the source of pollution, that makes managing and mitigating the pollution much easier. So with that, I would like to thank everyone who has supported this project, especially my PhD advisor, Dr. Jeffrey Turner, as well as everyone at the Hart Research Institute and the TGLO who helped with this project. And I'd like to thank the Texas General Land Office for providing funding for this project as well. And with that, I think we have time for a question and answer session. And if you have any questions, feel free to email me too. This is my contact information. You can just send me an email anytime if you're curious.
Thank you so much, Nicole. That was a fantastic presentation. And as though it was right on cue, it sounded like maybe a construction group got out right outside your window right at the beginning of your presentation, but you plowed right through it wonderfully. So thank you so much for that.
Yeah, sorry about that. There was somebody with a lawnmower.
No, it was as though it was on cue, but you did a fantastic job. Really packed with a ton of information here. Great presentation. There's so many different angles to go with, but we do have a couple of questions here. We'll take one on sea level and one about correlation with floods if we can. So Tom Hagemeyer asks, how much sea level rise has been measured along the Texas coast? Does this vary? What's the average rise?
So I will have to double check on those numbers, but I can definitely get back to you on that. We were using data collected and recorded by NOAA, and most of these locations did have a slight increase in sea level, but to get those specific numbers, I have the data on my computer. It will take me just a little bit of time to find it, but I can definitely send that information your way.
Yeah, it's an easy question to ask, right, Nicole? But it's a really complicated one to try to answer. As we're aware, based on different studies, that sea level does rise differently in different parts of Texas and in different parts of the world. The other piece is where are we taking these measurements? What equipment is being used, and how's that information being interpreted? It's almost like a separate standalone project all on its own. But we do use a network that we work, the Texas General Land Office through its TCOON network. We do partner with NOAA and U.S. Army Corps of Engineers to collect that sea level rise data. So there's quite a bit out there, but it's a very complicated question trying to get directly to the answer on. We do have a separate question from Ashley Jones, and she asks, what about correlations with flooding?
So that we didn't measure that during this study, but that could definitely have an impact. Flooding and just massive amounts of fresh water that's produced by storm events or hurricanes have been previously associated with really high levels of fecal pollution in the bay. So that's something that would be really interesting to investigate in the future too.
And if I could just add to that, when we were investigating the initial incident that raised our awareness to the very high values in Brazoria and Matagorda counties, the General Land Office looked in depth at the relationship between, at the time, we were at the tail end of a very large series of rains from spring. We had a very wet spring in 2019. And so we did look at the relationship at the time. We also looked at many of the smaller rainfalls that were falling in and around our monitoring sites and looked at the relationship between that runoff and the elevated values. And at the time, I mean, certainly none of us are going to say that there was no relationship, because we all know that stormwater impacts water quality, period. We were seeing very elevated values during dry weather periods, and that was something else that alerted us. So I see another question here, and this one's from Nick Dornack. Did HRI develop or acquire any geospatial data associated with septic systems in the coastal zone?
So we did have some septic system data that was given to us by the OSSF team at Texas A&M University. And we were investigating just the proximity in the counties to the coastal zone, the number of septic systems overall. And we saw a small correlation, and actually it was an inverse correlation between the number of the septic systems and Enterococci values in the Bayside sites. However, we think that data that includes the age and the type and the maintenance of the septic systems might be more informative in the future, rather than looking at just the number of septic systems.
Thank you, Nicole. It looks like I skipped over a question from Allison Buckteen in the Texas General Land Office, and she asks, are there any regulations that require local governments to improve these numbers? Are local governments alerted to these frequent elevated levels at their local collection sites? And I might take that one, and if you could follow up if you have anything to add. And typically when it comes to water quality, it is mainly regulated by the Texas Commission on Environmental Quality and through a partnership through the Clean Rivers Program and other cooperative data collection entities. And so ultimately the TCEQ has the ultimate regulatory authority here in the state of Texas in regards to ensuring that waters are swimmable. And so they have a methodology for doing the assessment, and then moving a water body, either, I guess, ranking it in different ways that would show that it's either meeting its use or not meeting its designated use. At the local level, there are many different interests, and all of them are about, everyone wants the best water quality that we can get. And so everybody who is in part of the Texas Beach Water Network receives notification for each of the monitoring events. And so we have local contacts with each jurisdiction, and there are many of them across the 169 stations. And so in many cases, the local jurisdictions are the ones who actually flip the beach watch signs or actually post that there is a swimming advisory there. It's also another one of those questions that can get very difficult to answer, because often, and it's really part of the underlying component of Clean Coast Texas, is that there's really no one entity that's going to be able to resolve some of these really complicated water quality issues. Often it takes a lot of science and investigation to try to just characterize and understand them. And so in many cases, it takes a team of people from different organizations to bring expertise and focus them for one goal, which is perhaps identifying particular issues and then remedying that particular issue. So we have another question here from SSA 56. Nutrients, temperature, and salinity are variables that are known to affect concentrations and the variability of bacteria. Are these variables being considered in your analysis? Nicole?
That's a really good question. Those variables actually were not considered in this analysis, but moving forward, we would like to definitely include metadata like that, as well as water turbidity and dissolved oxygen, because those variables definitely do play a role in how long Enterococci, might be sticking around in the environment. And that's going to be really important to look into in the future. But unfortunately, it was not included in this part of the project.
Yeah, that's it was pretty much at the case with any researcher in any project, in particular with one of this magnitude, is having the metadata or the data relevant to the bacteria observation. And there are literally, as Nicole mentioned, there's dozens, there's hundreds, if not thousands or more variables impacting the one main piece of data in this case, which is the Enterococci data. And so we're definitely interested in expanding the type of monitoring we can do on a routine basis. But what underpins that always is how do we manage that? How do we produce the money to try to manage a budget to implement that? And it's very expensive. And we're definitely interested, but currently have not been able to identify budget funds to get us to that point. Okay, and just as a reminder to folks, we're about 35 minutes into this event. We might end up going 60 minutes. If you have more questions or comments, please post them in the chat and we'll continue to review these here for the next perhaps 25 minutes if we have enough. Otherwise, I mean, I already feel we've been in this for 35 minutes and it has been packed with information. And I've learned a lot from your presentation as well. So we have Rhonda Gregg who has a comment. Matagorda County statistics are very interesting. Extreme erosion on Sargent Beach combined with sea level rise, and the beachfront is dotted with homes and aging septic systems. Again, values are very interesting considering the sparse human population and high bacteria counts. Very much worth additional investigation.
Yeah, so I've had a few people actually point out the interesting results in Matagorda County, saying they might not have expected it because the population there is a little bit lower than in some of these other coastal counties. And it's possible that this fecal pollution it might be coming from those septic systems, but it also might be coming at least in part from other animals that are present there as well. And so that's what I was saying about the bacterial source tracking, that would offer more insight into that hypothesis that animals are also contributing to the waste.
So part of the context for this discussion and the study results is also to look elsewhere to see, has this happened in other communities? Is this documented elsewhere as far as these relationships that have been identified through the study here? And I don't know if it's a lucky coincidence or not, but actually some areas in Florida have had some very similar occurrences, and in some cases along a similar timeline as us. I actually noticed someone from Florida Coastal Management Program is on the line right now, and we'd love to talk with you afterwards. We've been doing some coordination with them just to swap notes and to look at their perspective and how they've tried to tackle the issue. But it's certainly interesting that the relationship between our seas and septic systems and coastal water quality has been observed in a very similar phenomenon and coincidence to what we're seeing here. So we're definitely looking at our colleagues and peers in other areas to see where we have other conditions that we can learn from and then propel us into the next phase of the study. So we have a question from Juan. Do you plan on expanding the testing into populated coastal watersheds? And I'll go ahead and take that at least from a Texas Beach Watch perspective, in that we do have methodology for expanding our network into new areas that are being used. We typically are using what we call a tier 1 beach approach to develop these priorities. And what that means is in that county and in that jurisdiction, where are the beaches that are most heavily used? And we target those particular beaches. As some of y'all probably realize that over time, maybe some areas are not used as much where they were very heavily used in the past. So our approach is to analyze this information to see how the user patterns are changing, and then redirecting some of the monitoring to new sites when and if areas are becoming more heavily used and populated. We have another question here, and it is, they say the Dickinson Bayou waters are polluted. Is that important? And what can we do about that? If, you know, bacteria impairments are amongst the most prevalent reasons for impairments in the state of Texas amongst all the waters that are monitored through a vast array of partner networks. Thousands of monitoring sites and hundreds of different partners bringing that together. And so unfortunately, bacteria does happen to be one of the most significant ones causing impairments. I don't think we're able to speak specific to Dickinson Bayou, but I am aware of different water quality, watershed protection efforts going on in Dickinson Bayou. And if you would care to send an email or just a message in the bottom with your contact information, and we can follow up and get you connected. We have another question. Jason, you mentioned the budget challenges. Are there grants that can bridge the differences so cities can embark on major septic tanks to treated sanitary sewer system conversions citywide to improve the regional water quality? Yes, the answer, I mean, when you ask a question like that, how you convert from septics into a main treatment plant, that can be done and that has been done. There are different sources. You're kind of opening like a giant can of worms just to even start talking about the funding sources, but there are some fairly large funding sources available. And of course, when you look at this kind of conversion, I mean, it potentially could cost even $10,000 per home, depending on what the scenario is, and of course, building the infrastructure to support that. Another question from Nick Dornack. Were the counties identified as having the highest percentage of exceedances more a result of poor water quality or perhaps greater percentage of Baysides to Gulf sites?
Bayside and Gulfside sites could definitely be influencing that as well. So I'd have to take a look at the individual sampling sites and do another comparison to confirm that. But that's definitely contributing to the elevated levels, was because some of those sites were on the bay.
So we have another question about rainfall and Enterococci values. Did you include rainfall events and Enterococci presence when collecting data, besides in the sewage overflow?
So some of the data had rainfall that records and occurrences that accompanied it, but unfortunately not all of the data had consistent metadata. And because of that, we felt that it would be better to not do an overall analysis of rainfall on a big scale, because we didn't want to bias the analysis with only the data that had rainfall recorded, in case somebody might not have recorded it when there had been dry weather. So that's why that was not included in one of the overall objectives of the study.
In looking at rainfall and the relationship to our monitoring data with Beachwatch Network, it's super interesting when you end up looking at hundreds of these monitoring data points. And then, like, for instance, if you look up the Tex Mesonet data site, which is one of our sites we use for looking at precipitation, and that network of rain gauges along the coastline in some areas is quite prolific. And so you get some spatial acuity when you look at where the rainfall occurs and how that relates back to your one particular data point. And so what we found is a generality that's just anecdotal in its nature and approach, in that when we looked at the actual monitoring event and then tried to correlate that back to rainfall, what we found in the Gulf sites was that even if we're monitoring during a rain or immediately after rain, often the monitoring data, the stormwater runoff were not being identified in exceedingly high or even high values in many cases. Primarily we think just due to the large volume of water and the dilution factor. That combined with the very small watersheds, it's kind of odd to maybe even call it a watershed in some cases. These are just very small flow basins, if you will, and pour points that come down across these barrier islands and in the upland areas around bays. And so it's very difficult to tease out when you're looking at a monitoring site, and rainfall that may be just 4 or 5 miles away is not impacting that monitoring site. Let's see, we have another question here from Joel Behrens in Calhoun County. In regards to high levels in Matagorda, what about all the sanitary sewer systems that dump into the Colorado River and end up in Matagorda Bay, upriver from Matagorda County?
That could definitely be contributing to elevated levels of fecal pollution. That would be something interesting to look into further as well. Jason, do you have any more information on that?
And during the initial occurrence in 2019, we looked a lot at the flows coming down from the Colorado River, as well as the Brazos River, also from a couple of other smaller tributary streams, Caney Creek, as well as the San Bernard. And so many of those streams that I just mentioned are gauged in different locations. So we could look at the flow coming down from each of these. And we were not seeing, initially in our investigation, we were not seeing a relationship or a cause and effect, if you will. But one thing that's really complicated and kind of confounding to everything we're talking about here that we haven't mentioned much is what is happening to your drop of water in the beachfront. So everybody who's gone to the beach, and if you've gone into the water and waded out to the first sandbar, you know that where you enter the beach is not right in front of your umbrella. You quickly end up 20 yards down, 40 yards down, 100 yards down pretty quick. And so the same thing is happening with contaminants when they enter the beachfront environment. And so just because we're sampling one point doesn't necessarily mean the data are coming from that, or the contaminant we pick up are coming from that one particular point. And it's certainly a major confounding part of this discussion that's impacting our results and conclusions. Okay. So while we're waiting for a few more questions to come in, I just wanted to mention a couple of additional points. Earlier, I mentioned this is one of our engagement mechanisms for Clean Coast, Texas. We are currently seeking communities to partner with to figure out how we can engage in common interests, such as improving non-point source pollution and water quality in your community. This could be in the form of longer-term planning assistance. We've partnered with the Texas Community Watershed Partners out of AgriLife, as well as Texas Sea Grant and Texas State University as well. So we have assets and capabilities right now where we could work with local communities to figure out how we can try to solve common problems of interest. Along that lines, we have our next Lunch and Learn series. The second one will be in a month. These will occur approximately on the third Thursday. We're thinking about moving them to 11 to noon instead of noon to 1. The next one will feature Brian Sims, a senior planner at the Houston-Galveston Area Council. And for folks who may not be familiar with that organization, it's also known as HGAC, and it's in and around the Houston area, but its jurisdiction is quite expansive into numerous counties. This organization has been around for decades and has a tremendous group of expertise that they've brought to bear in their programs based on a lot of expertise and just a lot of experience in dealing with coastal water quality issues. So Brian Sims is going to share next month improving water quality through coastal community programs. And the idea of Lunch and Learn, and as well as in the next component, is what is being done elsewhere. While I didn't know about that, if we wanted to do something similar or explore something similar or even implement a program or a plan that's working elsewhere in our area, how do we do that? And so Brian is going to share some of that information from HGAC next month. And I see that, thank you, Leah, with Texas State University, Meadows Center for Water and the Environment has been posting a lot of links right here where you can get in and you can reserve yourself and, for the April Lunch and Learn. You can also get on the distribution list for future announcements. At the very top of the chat box, there are a couple of links. I didn't really talk much about the full breadth of the Clean Coast Texas program and why we're here and who we're working with and what we're doing. But if you want to learn more, we have our introductory meetings on YouTube where you can watch that meeting, 2 meetings that we already put up there a while back. And then of course, we're super excited about the brand new website, cleancoast.texas.gov, that is also available, which will be an important resource that's going to help tie our folks together we're working with along the coastline. Okay, I see we have another question here. How will the proposed opening of the mouth of the San Bernard River affect levels? And I could take a stab at that, and Nicole, if you want to fill in as well. The quick answer is we just don't know right now. And we're certainly very interested to see if and how there are any changes in what's already been observed and how those changes might be impacting future data results. Brian, thank you for posting that information there for introductory workshops. So it looks like the questions are starting to come to an end here. and everybody always appreciates a shorter meeting and getting out a few minutes early. Nicole, I'm super excited and super thankful. Your expertise in this study, along with Dr. Turner and Dr. Wetz, and Lucy Flores in helping us to pull together the study and the analysis is it's unprecedented for us here in Texas. You know, the Beach Watch program is really in place for one primary reason, which is to provide near real-time data to the beach patrons so they can look at that information and make a decision on their own about potential health risks associated with going to their particular swimming site or area they want to recreate. And so that's the primary purpose. But over time, in a program that's been operating for 20 years, we're now sitting on a massive treasure chest of information that we're just starting to dig into. And so thank you for, I would just call this step one, and better understanding and characterizing what's going on. And we'll have a lot of work in front of us here yet to do. Any closing comments, Nicole?
Thank you again for having me and letting me present on this research. This has been a really fun project, and I'm excited to keep working on it.
Excellent. So are we. I would like to thank and acknowledge Texas A&M University Meadows Center for Water and the Environment for facilitating this event and other events and doing a great job here in helping us to implement the Clean Coast Texas program. Like I mentioned earlier, we have a coastal collaborative with Texas Sea Grant, as well as Texas Community Watershed Partners in the Meadows Center, in which we are currently implementing program operations. If you're with a jurisdiction or with a local group who's partnered with the jurisdiction, please feel free to contact us if you think you're hearing things that resonate with you and your interest in your local community. And we're excited to get to work. Without any further ado, we want to thank everybody for joining us. We will be back next month and hope to see you then. Thank you so much.